The liver primary storage function in iron metabolism is to store roughly 1 gram of iron in ferritin and to mobilize iron rapidly in the event of increased consumption. The liver is also the source of the regulatory hormone of iron metabolism, hepcidin. The liver receives iron from the blood primarily via transferrin, an apotransferrin molecule that can bind up to 2 iron atoms. Transferrin saturation is sometimes given as percent of iron saturation, average from 20 to 55%. Reduced values in iron deficiency. Iron bound to transferrin enters the cells via receptor-mediated endocytosis. First, transferrin binds with iron to the transferrin receptors on the cell's membrane. The entire complex is then incorporated into the cell by the clathrin proteins to form endosomes. A proton ATP-dependent pump of the endosomal membrane pumps hydrogen ions into the endosomes, acidifying the contents. The reduced pH causes dissociation of bound trivalent iron from transferrin STEAP3 reductase, then reduces the released ferric ions to divalent ferrous ions. Ferrous ions and hydrogen ions are transported from the endosome to the cell cytosol by the DMPT1 transporter, similar to the DMPT1 transported on the apical site of enterocytes. In this way, iron from the blood is gradually transferred from transferrin to the free divalent iron pool in the hepatocyte. After iron is released from the endosome disappears and the entire apotransferrin complex bound to transferrin receptors is transferred back to the hepatocyte membrane. Free apotransferrin, that is iron-free transferrin, is released from transferrin receptors. Usually, almost all iron ions in the plasma are bound in transferrin. If the body is iron overloaded, non-transferrin bounding iron ions, referred by the abbreviation NTBI, appears in the plasma. These ions can be taken up into the hepatocytes by the DMPT1 transporter. However, the membrane enzyme ferrireductase must first reduce the trivalent ferric ions to divalent ferrous ions. They are then imported with hydrogen ions in the DMP1 transporter and absorbed in the cytoplasm. Iron is stored in ferritin primarily cellular iron stores, which is oxygenated to the trivalent ferric form from the cytoplasmatic pool of ferrous ions. The hepatocyte is a metabolically active cell. The DMPT1 transporter also transports iron into mitochondria which store iron in ferritin. The hepatocyte can also release iron from its stores. Iron is transported into the blood by ferroportin. Celluloplasmin, similar to hepcidin in enterocytes, oxygenates iron into a trivalent form that binds to transferrin in the plasma. Ferroportin is influenced by hepcidin, and increase in hepcidin decreases the release of iron from the liver. A decrease in hepcidin increases the release of iron from the liver. Two signaling cascades influence hepcidin formation in the liver. The BMP SMAD signaling cascade mediates hepcidin's response to changes in systemic iron requirements, in iron deficiency or iron overload. BMP6 knockout mice, however, retain the ability to increase hepcidin expression following inflammatory stimuli. The increase in hepcidin production by inflammation leads to iron sequestration, whereby the body attempts to prevent access to iron by infectious agents, most commonly bacteria. The consequence of chronic long-term iron is then anemia. The induction mechanism of hepcidin production by inflammation is mediated by increased interleukin 6 levels and by affecting STAT3 signaling pathway. In iron overload or response to chronic inflammation, hepcidin blocks iron release, and cells store iron in ferritin. Ferritin is also present in small concentrations in the blood, and its increased concentration is one of the indicators of iron overload, or, in the contrary, iron deficiency in the prelatent phase of iron deficiency anemia. Clarify the mechanism of iron regulation. Let's now play simulation games with iron metabolism model and review the following questions.